Good day, student. Welcome to the presentation for the school subject Biology Module 1 by the code DES Bio21. We are going to have a session where we are going to provide you with vital information that will help you in preparation of the upcoming examination for the year 2020. Let's start. The first thing that we need to look at is the define and the description of this characteristic of living organisms. It is vital to understand the 7 characteristics of living organisms, to know their meaning as well as the description. Case scenario can be presented to you where a diagram is given and you should be able to deduce what kind of characteristic is demonstrated by that diagram, for example. Now let's start with the 7 characteristics of living organisms. We have movement, respiration, nutrition, irritability, or sensitivity, meaning that you can either write irritability or sensitivity, growth, excretion, and reproduction. Now let's look at the meaning of each of these words. So meaning the definition. The movement is an ability for an organism to change position using locomotion organ. So an organism must be able to change position. Movement in animal is very obvious, while in plants it's not obvious, but plants do change position where their leaves, their shoot, or their root change position. And that changing of position, it is a characteristic of movement. Now take note, living organisms must move on their own. It must not be with the help of another organisms. They must move, they must change position on their own. Let's look at respiration. Respiration is a releasing of energy from food substances inside the living cells. Respiration occurs inside the cells. There is some confusion between the respiration and breathing, please. Breathing is about the inhaling of oxygen-rich air and exhaling of carbon dioxide-rich air. It is not the same as respiration. Respiration only focuses on the releasing of energy, and it occurs inside the cells in an organelle called mitochondria. Moving on, and next, the third characteristic of living organisms is nutrition, which have to do an ability for living organisms to take in organic and inorganic nutrients. This definition applies to all living organisms. For example, the plants. The plants are able to take in inorganic nutrients such as water, carbon dioxide, as well as light, which they are going to obtain energy for growth and also to make organic nutrients. On the other hand, the nutrition in animal, the ability to take in organic nutrients, which were produced by plants. The key word there, nutrition, please, an ability to take in. It's not about feeding, because that characteristic must accommodate all living things. It's the ability for living organisms to take in organic and inorganic nutrients, from which they are going to obtain energy for growth, tissue repair, movement, and to carry out any other activities. The next characteristic is irritability and sensitivity. Sensitivity or irritability is an ability for living organisms to detect and respond to the stimuli. Living organisms, for example, animals, they do have sensory organ. The sensory organ, inside the sensory organ, they are receptors that detect a stimuli, and take note, a stimuli is any sudden changes in the environment. Now, once the receptor detected stimuli, then that organisms have either the effectors, and the effectors here we are referring to the muscles and the gland that respond to the stimuli. Now, take note, sensitivity and ability for living organisms to detect. There is a detection first, and then after they detect, they respond to that stimuli. Either to be quiet if the stimuli is pleasant, or to move away if the stimuli is harmful or dangerous to such living organism. In plant, we can clearly see how a shoot of a plant, especially when you put the seed in the soil and the seed germinate, and the seed first grow into a plumule and later it grow into a shoot. That shoot, either depending on the amount of light and where it's coming from. If it's coming from one side only, you will notice that that shoot will detect light and it will respond by growing towards the light. So very, very important there. In animal, it's very obvious. Obvious. We see animal detecting to the sound, to different stimuli, and they detect and they respond to it, either by moving toward it if the stimuli is pleasant, or moving away from that stimuli. Let's look at the next characteristic, growth. Growth is a permanent increase in the cell size and number. So living organisms must be able to grow. And that growth we are looking at in terms of number of the cells and also in terms of the cell size. The cell either increase in number by two types of cell division called mitosis or meiosis. Let's look at the next characteristic. The next characteristic is excretion. Excretion is different from ingestion. When we are looking at excretion, we are referring to the removal of waste product of metabolism. Majority of students do not understand that. Not all waste products are excreted. Only those that are produced inside the cells. And those waste products that were produced inside our cells, we call them waste product of metabolism. And those are the only one that we excrete. So, of course, a human being is we see a person going to the toilet and defecate. The defecate is not excretion. That is ingestion. Why it is not excretion? Because the feces were never part of the cells. Those are the leftover, undigested food from our alimental canal, plus bacteria. So, they reach the rectum and then we expel it out. But what we excrete out, please students, they must be waste product of metabolism, meaning that they are waste product produced inside the cell. We don't only excrete waste product of metabolism, but we also excrete substances that are in excess than what the body required. For example, you take in more water. Excess water in your body need to be excreted because it is in excess than what your body required. Let's go back to that definition again. Excretion, it's a removal of waste product of metabolic as well as substances that are in excess than what the body required. Let's move on to the next characteristic. The last one, reproduction, is the ability to produce offspring. When a living organism produces offspring, we call it reproduction. Either asexually, where you only involve one parent, or either sexually, where there is involvement of gametes coming from a male, one coming from a male and another coming from a female. Therefore, in sexual reproduction, you have two parents involved. Very good. Let's go to the next. The next thing, we are going to look at kingdoms and their main diagnostic features. There are 5 main kingdoms. The 5 kingdoms, we have prokaryote. Prokaryote is also called kingdom Monera or kingdom bacteria. These are all the bacteria are classified in kingdom Monera or prokaryote. Remember please, the kingdom prokaryote or Monera or bacteria, they are unicellular. And one major difference between them with the rest of the kingdom is that they don't have a nucleus. Now, their genetic materia are just floating inside the cytoplasm. They do have a cell wall, which is made up of peptidoglycagon or murine. They also have a cell membrane, a cytoplasma. They do not have any membrane-bounded organelles. For example, the mitochondria. For example, the chloroplast. They do not have any organelles that have double membrane. Please take note of that. So you need to be able to understand the main features for the kingdom Moneras. So another kingdom is Protoctista. The kingdom Protoctista is one of those you find majority of these animals or plants, they are not really specific animal. Neither plant, they are at a borderline. Some of them look like animals, some of them look like plants. We have, for example, amoeba, we have euglena. Euglena, for example, it contains chloroplast, meaning that it photosynthesizes. Now, the Protoctista, majority of them, they have a nucleus. But you still find some that do not have a nucleus. Another thing to pay attention to it is that they do have certain features that are more at an advanced level compared to the kingdom Monera. Please, there are quite good information in your modules there. Please focus on this also kingdom. The next kingdom that I would like to look at is kingdom fungi. The kingdom fungi, they are not plants. Yes, they resemble plants in a sense that they have a stem-like structure, a root-like structure, but they are not plant in a sense that they lack chloroplast. They are not green, so they cannot photosynthesize. So they do have a cell wall made out of chitin, and they do not have proper roots, neither they do not have proper stem. Therefore, they are not classified as kingdom plant, but kingdom fungi. Their body are made up of hyphae. So please pay attention there. The next kingdom, the 4th kingdom is kingdom plant. Plantae or plant. Plants are very prominent. They do have chloroplast and they are photosynthetic organisms, meaning that they produce food. They have a cell wall made up of chitin. They do have the vascular bundles that made up the transport system. They are all multicellular, meaning that they are made up of many cells, and they are all eukaryotic organisms because they contain a nucleus. Now, we can look at in terms of the difference between kingdom fungi and kingdom plants. Kingdom fungi, they are cell walls made up of chitin, whereas kingdom plants, they are cell walls made up of cellulose. Now, another thing, the last kingdom is kingdom animals. The animal is more advanced in terms of the cells, tissue, organ that they have. The animal, they are made up of nerves, they do have muscles, they have a cell membrane, but they do not have a cell wall. Now, there are many animals that are found on earth. Some that are classified, some that are yet to be classified. So it's very important to look at those four. We also, looking at the virus, we didn't mention any kingdom where you can place virus. Virus is not placed neither in these 5 kingdoms, because virus still possess some features of living organisms, and it also possesses features of non-living organisms, meaning that it contains, for example, genetic materials. It can reproduce, but it does not possess any other features that can classify it as a living thing. For example, it's made up of protein coat. It doesn't made up of cell wall or cell membrane. It does not have a cytoplasma. It just have a core. So you should also be able to look at the difference between these kingdoms. Let's talk about the next phylum, the phylum Arthropoda. We have 4 main classes within the phylum Arthropoda. We have insect, arachnid, crustacean, myriapods. Let's go back to the arthropod. Arthropods are organisms that are invertebrates. They have segmented body, meaning that their body are divided into parts. They have jointed legs. Also, they do have a pair of legs. They have pairs of legs. Now let's talk about insect, arachnid, crustacean, myriapod. The insect, they have 3 body sections: head, thorax, and abdomen. Also, they have a pair of antennae. They have also compound eyes. We also look at the 3 pair of jointed legs, which are located on the thorax. We go further to in terms of the wings. Some have wings, some don't. We go to arachnid. You need to look at the diagnostic features of arachnid, the crustacean, and myriapods. Myriapods, where you have 2 subclasses, where you have millipedes and centipedes. Now, the unicellular and multicellular organisms. Unicellular in biology, when we are referring to uni, means 1, meaning that living organisms is only made up of 1 cell. Example, we have bacteria. Multicellular organisms are organisms that are made up of many cells. You and I, the plants, the kangaroo, the chimpanzee, all those. We look at the dichotomous key. The dichotomous key, you need to look at the definition of it and how to use it. It's one of artificial way where we can identify unknown organisms. Biological drawing, it's in grade 12, grade 11 for the new curriculum. There is always drawing in paper 3, question paper, or paper 2, ordinary level. Now, you need to make sure you know when you are given a drawing and you need to draw your drawing, you need to pay attention to the outline. Outline, we are looking at the line quality. We are looking in terms of you should not have any shading on your drawing. Looking at the proportional, if you are given an insect with 3 pair of legs located on the thorax, your drawing should also have 3 pair of jointed legs located on the thorax. The proportional means your drawing must look exactly like the one that is given. Labeling, your drawing must be labeled. Let's say you are given a diagram of fish that you have to draw. It must have scales, fins, and an eye. Let's look at the structure of the cell membrane. The structure of the cell membrane is a part of a cell. You need to know the hydrophilic, the hydrophobic, the hydrophilic referring to the head, the hydrophilic referring to the tail. You also need to look at the protein that are found in the cell membrane in terms of channel protein, in terms of carrier protein. You need to know and understand it. Let's talk about the movement of substance, how substances move in and out of the cells. Substances move in and out of cell by 4 different processes. Diffusion, which is a movement of substances in and out of the cell from a high concentration to the low concentration, down the concentration gradient. Please, diffusion, we are referring to the movement of particles, any particle, gas, as long as those particles are moving or die from a high concentration, meaning that from where they are more to where they are less. We look at facilitated diffusion. Facilitated diffusion, we have a movement of substances across the cell membrane through channel protein. So again, these substances are moving from a high concentration to the low concentration. The difference between diffusion and facilitated diffusion is that diffusion, the molecules are moving through the cell membrane through the phospholipid, meaning that they are not entering the cell or leaving the cell through the protein. They are passing or entering the cell through the hydrophilic bilayer. So as long as the arrow is moving to the cells and it's moving across the hydrophilic layer and it's moving from a high concentration to the low concentration, that is diffusion. On the other hand, if the molecules is moving from a high concentration to the low concentration and it's by channel protein, then that is by facilitated diffusion. Osmosis has to do with water. And we say it's a movement of water molecules from a high water potential to the low water potential through a partially permeable membrane. In active transport, you must refer to the ATP. It is the movement of molecules from a low concentration to the high concentration with the aid of ATP, meaning that it requires energy release from ATP there. The type of solution refer to a liquid where you have solute dissolved in. Now in biology, any hyper always means more, meaning that I have a container of water and I put 10 teaspoons of sugar. That solution, because I put 10 teaspoons of sugar and I have another container and I only put 2 teaspoons of sugar, where I put more sugar, that solution is called hypertonic, meaning that it's a solution that have more solute than water. Hypotonic, it's a solution that have less solute, meaning that, for example, the example that I gave where you have a container and you put less sugar, that solution is hypo. Hypo refer to less solute and hyper means hypertonic, in this case refer to more solute. And let's talk about the isotonic. Isotonic is where the solution, that liquid where you place your cell, either a plant cell or animal cell, has the same solute, meaning that the cytoplasm of the cells or the vacuole of that cell, if it's a plant cell, it has the same solute as the water that surrounds it. So there will be no osmosis that will take place. If the osmosis will take place, it will be very slow because the solute are equal. Let's look at the enzyme. Enzyme are protein that act as biological catalyst. You need to look at the definition of enzyme. As I said, protein that act as biological catalyst. The type of enzyme, the scientists that have done extensively work in project related to enzyme. We look at the photosynthesis in terms of definition, and also in terms of equation, both in words, and you really need to be able to balance a chemical equation of photosynthesis. Let's look at the leaf structure. The leaf structure, we look at the external and the internal part of the leaf. The external and the internal part of the leaf we are looking at in terms of, when you look at the leaf blade, the leaf apex, the leaf stick, you should also look at the leaf surface, the layer of it, and how is the features, the external features is enable the leaf to be able to absorb maximum light needed for photosynthesis. Now when looking at the internal part of the leaf, we have the cuticles there that is made up of the oil and it's reducing transpiration there. We also have the upper epidermis on the upper side of the leaf. The upper epidermis is very, very important to pay attention to. They do not contain any chloroplast, and this is important so that it only ensures that maximum light enter to ensure that more photosynthesis occurs inside the cells. And also from the upper epidermis, we do have parasite cells. The parasite cells there, they are rectangular, they are located on the right angles, and they contain numerous chloroplasta. And the numerous chloroplasta absorb much light And this is where most photosynthesis takes place. Below the parasite cell, we have spongy mesophyll cells. And between the spongy mesophyll cells, we do have air space. Now, the spongy mesophyll cells also contain chloroplast, but not as numerous as in parasite cells. Meaning that in a spongy mesophyll cells, we do also have photosynthesis take place. The air space ensures that the diffusion of carbon dioxide and oxygen, diffusion of carbon dioxide into the cells, and carbon and oxygen out of the cell. Take note, carbon dioxide diffuses into the cell during photosynthesis because it is a raw material of photosynthesis. Good. Let's look at the experiment on photosynthesis. There will always be some experiment on in biology. It's a practical subject. Now, the idea here is you want to find out whether light is needed for photosynthesis. If you put your plant in a dark cupboard, in a dark room, would it photosynthesize? So you need to look at some of the experiment. The best way how you do it, you need to remove the best experiment to use is a potted plant. You remove it from the sun or where it have access to light, and you discharge it. Discharge it means you put it in an area where it's in the shade or inside the cupboard so that the starch that is there can be used up by the plant. And once it's used up by the plant, that plant, you can now, after 3 days perhaps, you take out the leaf, you boil the leaf to soften the leaf and to destroy the cell wall, the cell membrane, and you put the leaf in the test tube with a methylated spirit. You boil the methylated spirit in the water bath. The idea is to remove chlorophyll. And once you remove chlorophyll, you then you have now a white leaf. Then you put it on the tiles. After putting it on the tiles, you add iodine solution. And if that leaf turn blue-black, that means starch is still there. So that means you need to continue putting it in the dark room. And finally when you come and test it and you notice that the leaf turn yellow, then there is no starch. So that can come to the conclusion that yes, you need light for photosynthesis to take place. The process of carbon dioxide, very simple. You can have two plants where you have a presence of carbon dioxide, and another one you cover the potted plant and you put a substances that will absorb carbon dioxide. And then you test these two after, for example, in 24 hours, you test the leaf for the presence of starch, meaning that you use iodine solution. Presence of chlorophyll, very easy again. You use two leaves. One is green, meaning that it contains chlorophyll, and one is yellow, meaning that it does not contain chlorophyll. And you test for test for starch. Once you remove the chlorophyll and you put iodine solution, you are expected to see the following result. The green leaf turning blue-black, the yellow leaf, it will remain blue, meaning that chlorophyll is needed for photosynthesis. The same as with water. And test for carbon dioxide, please bubble through clear lime water and it will turn milky. Now, the presence of carbon dioxide, again, use a glowing spleen and it will relight. Good. Human body, please look at the breathing system, the nose, the trachea, the bronchi, the bronchiole, the structure of the alveoli in human body, lymphatic system, and the lymph nodes, the immune system, phagocyte, the antibody production. Now, if you have any inquiries regarding any content that you read through your DES, Bio21, the information on your screen, the email address is there, and the telephone number, I will be of your assistance. Thank you.